Welcome to this session on the course Organic Chemistry in Biology and Drug Development. Okay. We have, as you have noticed that towards the end, these last few lectures combined biological pathways with the discovery of drugs. So, some medicinal aspect we are already into some medicinal aspect of whatever the chemistry that we have learned while studying the biological system. Okay. Examples are this folic acid biosynthesis inhibitions. Now, next coenzyme that we are going to discuss is what is called vitamin K. Now, this K actually stands for coagulation, but it is a in German language this starts with a K. So, that makes it that this is vitamin K. It is coagulation means it is a blood coagulation, that means which is involved that whenever there is a cut, we know that after few seconds or minutes the blood won't come outside. So, there is some clot, we said some blackish thing that covers the cut, and that is what is called clot, and this process is dependent on the vitamin K. So, if you are deficient of vitamin K, if you have a deficiency vitamin K deficiency, then blood clotting will be a very slow affair, and so lot of blood will be lost. In addition to vitamin K, also you need calcium, lot of calcium ions, because the clot is made up of a protein which is called fibrin or fibrin. This fibrin protein in presence of calcium forms cross-linking of proteins, and that makes its very insoluble mass, which is known as the clot. Now, how this vitamin K is helping to form the clot, we are going to decide that, we are going to describe that. Okay. Now, blood clotting process is actually a large number of cascade reactions. Cascade reaction means suppose A goes to B, and then B goes to C, and then maybe C catalyzes the reaction of D to E. So, like that E catalyzes the reaction of F to G. So, these are called cascade reactions. So, there are a large number of cascade reactions. Total number is that in which a dozen or so serum glycoprotein function catalytically, and subsequently with the ultimate conversion is that formation of fibrin. That is the last step in blood coagulation, and fibrin in presence of calcium forms the cross-linked fibrin, which is called the clot. Now, what is the starting material for fibrin? It is what is called fibrinogen. Fibrinogen is a larger protein, as compared to fibrin. So, at the time of this cut, ultimate reaction is that fibrinogen has to be converted to fibrin. So, it is a very large protein. So, you have to divide it into two. You have to take out some portion from the fibrinogen to make the fibrin. Okay. So, basically what happens, if this is fibrinogen, then you cut here and you get the fibrin. Okay. And this is fibrinogen, and this is true for many of these proteins or enzymes that are present in our body. Like when you take food, that food will be digested by chymotrypsin, trypsin and pepsin, and this chymotrypsin or trypsin actually are present in an inactive form called chymotrypsinogen. So, as soon as I take the food, the system gives a signal that chymotrypsinogen has to be broken down into the active chymotrypsin. So, that is nothing but a proteolytic cleavage of one portion in the long polypeptide. So, you take out some of the portion which is making it inactive. So, that portion is chopped up and you resulting in the active form of the protein. Okay. So, that is but this breakage is nothing but a protease reaction. You are basically breaking the peptide bond. So, and that is peptidase or protease. So, this fibrinogen to fibrin conversion is catalyzed by a protein which is called, or an enzyme which is called thrombin. And now the question is, who prepares the thrombin, who synthesizes the thrombin? It is something called prothrombin. Prothrombin, by the action of many species, like factor 10 is a protein, factor 5 that is also a protein, these are all glycoproteins, and then calcium and phospholipid, all these are present. If all these are present, then prothrombin goes to the thrombin, the actual enzyme which catalyzes the conversion of fibrinogen to fibrin. Okay. Now, before that prothrombin, there are other, the previous cascade reactions, because I told you there are at least a dozen of cascade reactions. These are the final steps of blood coagulation. Prothrombin goes to thrombin by the action of this factor 10, factor 5, calcium and phospholipid, and the thrombin that is formed, that hydrolyzes fibrinogen to fibrin, and fibrin in presence of calcium forms a cross link. So, we are going to discuss this step where vitamin K has a role, prothrombin to thrombin. Now, what is that? Let us see. Prothrombin is formed initially, I can again go back to the slide. So, when we had the prothrombin, before that there is what is called immature prothrombin. Immature prothrombin. So, let me erase those. So, this prothrombin is generated from immature prothrombin. What is immature prothrombin? Immature prothrombin is that this, if you look at the protein, the amino acid sequence in this protein, so you will see that there are many glutamic acid residues at the terminal end. So, there are at least 10 glutamic acid residues within a stretch of 1 to 40. That means, this is say 40 amino acid residues, out of it 10 are glutamic acids. Okay. Now, that is what is prothrombin, and in thrombin what happens, this immature prothrombin, all the glutamic acids, this is the glutamic acid. So, it says that immature prothrombin has 10 of the glutamyl residues in the first 42, not 40, 42 residues from the amine terminus. Okay. From the amine terminus you have this. So, amine terminus, and then this is the immature immature prothrombin, and this is, if it is this 42 in number, then you have about 10 glutamic acid residues, and these glutamic acid residues are modified into gamma carboxyglutamyl residues. That means, glutamic acid is already having this is alpha, that is beta, and this is gamma. So, it has already the carboxylic acid as the side chain, but in addition to that, this in presence of vitamin K, this one of the hydrogen is replaced and you form another carboxylic, you introduce another carboxylic group, acid group, and they will be present as both as minus, and that makes a good complex with the calcium ion, and this is what is called the mature prothrombin. Mature prothrombin. So, difference between immature and mature is that in immature prothrombin, all it contains a large number of glutamic acids, and in mature prothrombin, these glutamic acids are again further gamma carboxylated with the help of vitamin K to form the gamma carboxy glutamic acid, okay, or gamma carboxyglutamyl residues. Why you need these gamma carboxyglutamyl residues? Because they form complex with the calcium, and that is a necessary condition for the prothrombin to work to carry out the next reaction. Remember, what is the next reaction? this prothrombin goes to thrombin, okay, goes to thrombin. So, unless this carboxylation happens, this immature prothrombin cannot go to the cannot do that. That means, it cannot be converted into the thrombin. Okay. I hope that is more or less clear. Let us see what is the structure of vitamin K. Vitamin K has this quinone moiety and attached to what is called a phytyl side chain. It is a C20 side chain, phytyl chain, okay. C20 made of C20, 20 carbon atoms. Okay. So, this vitamin K, this is the oxidized form, a quinone, and in the reduced form it is a hydroquinone, that you can have OH and OH here. Okay. The carboxylation process is vitamin K dependent and it acts as a cofactor for the carboxylase. So, let us see what is the mechanism of that reaction. Again, just to remind you quick that what we are discussing is how these glutamic acid residues are gamma carboxylated with the help of vitamin K as the coenzyme. Now, the mechanism that is the most accepted mechanism is what was proposed by Paul Dowd, a famous organic chemist, and this is what is the proposal, and there are a lot of proofs basically are also forwarded when proofs are given, many evidences to show that this mechanism is possibly the correct one. So, what happens here? First, this quinone form of vitamin K by an enzyme called quinone reductase, it goes to the quinol. It goes to the quinol state, and then quinol means it becomes a, again this is an electron withdrawing system and this is the electron donor system. Okay. So, now this lone pair, what happens? This lone pair can come here and this goes here and that attacks the oxygen and this goes here. Okay. So, initially what you will get is this. See, when in the earlier mechanism before 1980s, another mechanism was proposed, but that did not involve oxygen. Everybody thought that the oxygen does not play any part here, but later on it was found that oxygen is required to do this carboxylation, and then Paul Dowd did some experiments and to show the involvement of oxygen in this way. Okay. So, what is happening here? So, you have this double bond O and then a double bond here and you have a OH here and this will become OO minus. Now, you can say that actually 2 electron transfers are prohibited in case of oxygen. The normal oxygen is in the triplet state. So, you can do the mechanism by single electron transfer, you will get the same thing. So, correct to be precise, this should be triplet oxygen. So, this type of transfer should be 1 electron. 1 electron means like this. Okay. So, but ultimately you will get either here dot and a dot here, and in case of this ionic mechanism you will get O minus. So, that involves singlet oxygen. So, if there is any singlet oxygen, then you can show this mechanism. I am showing for that with the assuming that this is in the singlet state. So, this can come back. So, there are 2 substituents, remember this is R, that is the phytyl chain, and this is the methyl, and that goes here and this goes. So, basically that is what is written here, this intermediate, this dioxane intermediate. So, basically it is a 4 membered ring with 2 oxygens, okay. Dioxytane, actually this is called dioxytane intermediate. And then after this what happens here is that this comes again back, let me see, this goes here, the mechanism is not shown in this case. I will just take few minutes. If this comes here, so you get the original state what I have showed to you. Okay. So, to make the epoxide, what is happening is this rather than going there, this attacks the oxygen because the OO bond is unstable. that can easily break. So, instead of going to this, because if it goes here, then you will get the previous intermediate. So, now it is attacking this oxygen, one of the oxygen in the to break the oxygen bond, the peroxo bond, which is very weak. So, you get this another intermediate where there is an epoxide here, and this is O minus, but this O minus is what is an alkoxide. So, that will be extremely basic, alkoxides are extremely basic. So, this alkoxide now can abstract the hydrogen at the gamma position of the glutamic acid residues. Okay. So, all these are required because this hydrogen has to be abstracted, and that requires a strong base, and this through this oxygen mediated pathway you get this vitamin K epoxide and an alkoxide. The alkoxide now takes the hydrogen from here. So, it becomes OH, and you know 2 OHs in one carbon means basically it is a carbonyl. So, that goes to carbonyl with loss of water, and this becomes the glutamic acid residue becomes the anion, and this anion now reacts with carbon dioxide, and that is how this carboxylation takes place. Okay. Now, this vitamin K is now basically in the oxidized form, and moreover it is having a epoxide here. So, this is called vitamin K epoxide. Now, in order to have the second cycle of reaction by the same vitamin K molecule, so you have to reduce it now. So, that is called vitamin K epoxide reductase, and you get first vitamin K quinone, that means this one, and now these enzymes are already there. So, the quinone reductase now will again take it forward to perform the next cycle of reactions. Okay. So, vitamin K epoxide reductase, so that takes the oxygen off and puts a double bond here. This is very important enzyme, because if it is inhibited, then what will happen, your next cycle of reactions will not take place, and ultimately the clotting that will affect the formation of the matured prothrombin, and affecting of matured prothrombin is a problem, because that will not be converted to if it is immature prothrombin, that will not be converted to the thrombin, and if there is no thrombin, then fibrin cannot be formed from fibrinogen. Okay. So, there are molecules which are called anticoagulant. Anticoagulant means that it actually does not allow the blood to clot readily. Okay. Now, you might say that for what reason we want to stop the clotting of blood, that is so required if there is a cut. It is required for people who are having who are suffering from hypertension due to the narrowing of the blood vessels. Okay. So, if the blood vessels become narrow, then what happens, that there is a lot of stress that is involved in the movement of the blood. So, if you can make the blood not to clot, because clotting means basically the stages of clotting means you are increasing the viscosity or density of the blood, and if you can stop this coagulation process, because many of the strokes, the heart attacks, they are basically catalyzed by this formation of the clots, because they are precipitates, they are solids. So, they can block the valves in the heart, or if they can go to the brain, then that calls the cerebral attack. So, you need what are called blood thinners, that means that allow the blood not to clot, and there are compounds like dicumarol, and there are compounds like warfarin, the structure is shown here. They are used as this is blood thinner, but they can be used also to kill the pesticide or insecticide. Basically, warfarin is a rodenticide, means rodents like this, the mouse, the mice and all these things which destroy the crops in the agricultural field, or in household you can use warfarin to kill those the rodents. Okay. So, rodenticide, and best is rodenticide is there are a lot of big grasshoppers, and they cause lot of problem with the production of they can destroy the agricultural field. So these grasshoppers, for that grasshopper, to prevent that attack, you can use warfarin. So that is one type of use. The other use is basically people who are prone to heart attack or cerebral, they use this dicumarol or warfarin, and even they also use aspirin as a blood thinner. Now these compounds, the dicumarol and warfarin, they work by as inhibitor of the epoxide reductase, that vitamin K epoxide reductase inhibitor. So it inhibits that enzyme and cutting of that catalytic cycle. Okay. So that is the called that's why they are anticoagulant drugs, very effective, very important. Okay. Next, vitamin chemistry. So vitamin K is over now, and many of the vitamin B complexes we have covered, vitamin B1, thiamine, pyrophosphate, the coenzyme form. Then we have the we discussed the vitamin, the pyridoxal phosphate, we have discussed folic acid, we have discussed some part of the flavin adenine dinucleotides, and we have also we have discussed the lipoic acid biochemistry. So the next one is what is called biotin. Now biotin was originally called vitamin H, but now it is again grouped into the vitamin B complex, vitamin group of vitamin B. Okay. Now biotin, the structure is shown here. The biotin is basically a bicyclic compound, and this is a urea like moiety, NHCONH, a 5-membered ring. This is 1,3-triazole, but in the saturated form, and this is what is called a thiophin, but tetrahydro thiophin moiety attached to a long chain carboxylic acid. Okay. So that is biotin, and in the actual so this is the cofactor. Interestingly, biotin and vitamin K, if you look at the structures, vitamin K, that is the quinone form, has to be reduced in order to form the coenzyme, and in this case biotin, it is the biotin itself which is attached to the enzyme as a prosthetic group that can itself act as the coenzyme. So you do not need any transformation as such. Okay. If it is given from outside, then it will be attached to the enzyme via a lysine moiety, because this is a carboxy. So you make a peptide from here, an amide bond to the lysine, and then this will be called a prosthetic group. Okay. So this is the enzyme bound biotin. So what type of reaction it participates? In what type of reaction it participates? Now this is very important. Again, all the vitamins are very important because they lead to these coenzymes, and coenzymes are responsible for various metabolic activities in the body, which are absolutely essential. Okay. Now the biotin is again, it does carboxylation. Okay. Like your vitamin K does carboxylation, but the role of vitamin K is to form this alkoxide in presence of oxygen and remove the hydrogen from the gamma position of the glutamic acid. So that is a separate class. Now we are talking about biotin. What biotin does, it participates in reactions like this. Like if you take pyruvate, and again the source of carboxylation is carbon dioxide, it could be in the form of bicarbonate, and in presence in case of biotin, it is in the bicarbonate form, and in presence of ATP, what happens that it forms this CH2 CH3, one hydrogen is replaced by CO2, because that is what is carboxylation. So you get what is called oxaloacetate. This is the oxalyl group, and so you get, and this is the acetate moiety. So you get oxaloacetate, plus what you will get? You will get ADP and a phosphate. That means ATP is hydrolyzed. So the energy of this is utilized to do this reaction oxaloacetate. Okay. And then acetyl coenzyme A, we have read the formation of the acetyl coenzyme A by the thiamine pyrophosphate, lipoic acid and FAD cascade of reactions. So, bicarbonate and again ATP. So, this will what it does, it again does carboxylation here. So, you have a carboxylation here, that is called malonyl coenzyme A and then ADP plus a phosphate. This malonyl coenzyme A is also a very important metabolite for making the different fatty acids in our body, that is obtained from malonyl coenzyme A together with acetyl coenzyme A. Okay. So, the mechanism, what is the mechanism of this reaction? Biotin is your NH and NH, but you can write the enolate form. Basically, if there is a base, the base can abstract the hydrogen, it goes to the enolate. Okay. And the enolate, when it comes back, remember you have this ATP. So, now the ATP, the first reaction that happens is ATP plus your bicarbonate and that goes to ADP plus this species. This is what? A phosphate linked to a carbonate. Okay. A carboxy, basically this is what is carbonate. So, you can call that as a carbonate, but if you call it as phosphate, then you have to say it is a anhydride of phosphoric acid, mixed anhydride of phosphoric acid and carbonic acid. Okay. So, now what will happen, this being an electrophile and the negative charges are taken care of by magnesium. So, that can come and attack the carbon in the carboxyl, this carbon in the carbonate species and forms this O minus, and then that is attached to this O phosphate and magnesium, and what is the next reaction that comes here and the phosphate goes out. Okay. So, basically earlier ADP was there, now phosphate goes. So, what you get is carboxybiotin. You have N-carboxybiotin, that is the intermediate. Now, from N-carboxybiotin, what you will get is that, suppose you have this, so N-carboxybiotin is N double bond O, then NH, then this sulfur and that. So, you have this CO and O minus N-carboxybiotin. Now, obviously, this will be has to be taken care of if you want to attack by a nucleophile. So, that is the chain attached to the enzyme. So, now your acetyl coenzyme A, SCOA. So, now there is the base in the enzyme that takes up this goes here and that is kicked out. Okay. So, you get carboxylation alpha to CH2 COA, CH2CO SCOA, that is CH3. So, we have written the CH here just to show the abstraction by the base. So, base abstracts the hydrogen and that goes here and this comes out. This is a very good leaving group because the nitrogen now negative charge is stabilized by the carbonyl. So, you get carboxylated acetyl coenzyme A. So, which is basically nothing but malonyl coenzyme A. A very similar reaction will be there for conversion of for formation of the oxaloacetate from pyruvate. I think you will be able to draw that. Okay. If you want me to draw, it is the same mechanism. You first form the carboxybiotin via the mixed anhydride of carbonate and phosphoric acid and carbonic acid and phosphoric acid, and then you have a nucleophilic attack on the carbon forming the carboxylated biotin, and then that carboxylated biotin, it releases the carboxy group and it carboxylate the alpha hydrogen in acetyl coenzyme A, and in this case it will carboxylate the hydrogen beta to the carboxy. So, this is alpha and this is beta, this is alpha keto acid. So, that is the beta carbon. So, the beta carbon hydrogen is also very acidic. So, that will be abstracted and instead the carboxy group will be put on the beta carbon, and releasing in all these reactions the biotin is again released, and biotin can again participate in the next cycle of reactions in a similar way. Biotin, there is a protein called avidin. The avidin and biotin complex is one of the strongest complex in, means biotin is the substrate. Of course, biotin is a substrate and avidin is the protein. So, protein substrate complex, that is one of the most stable protein substrate complex that you can get. So, if this has been utilized for biotin is utilized for affinity chromatography as a medium for affinity chromatography, because it has very high affinity to. So, if you attach a molecule to biotin, I can just explain very quickly, and then this molecule interacts with some enzyme, then you can by avidin add avidin. So, if it is in presence of various other proteins, so only that protein which has the small molecule attached to biotin, that will come out, that will be pulled down from this mixture of proteins. So, that is called affinity guided separation. Okay. So, that is the utility of biotin in protein purification, but here we have talked the utility of biotin in as a cofactor. Okay. And this again I remind you, this malonyl CoA, if it is extremely important, because that is the starting point is acetyl coenzyme A, but then part of the acetyl coenzyme A is converted to malonyl coenzyme A, and then they do react with each other and form a 4 carbon acid, and then that slowly increases by 2 carbons, and ultimately palmitic acid or stearic acid or oleic acid are formed. Okay. That is the biosynthesis of fatty acids. Okay. So, that takes care of the another B group of cofactor, that is biotin. So far, so we have the B group, we have discussed TPP, we have discussed PLP, we have discussed the FAD, we have discussed lipoic acid, we have discussed folic acid, now we have discussed biotin. Okay. So, next remaining is what is called cobalamin, that is the B12 vitamin B12 group. Okay. So, thank you.